So I want to take a moment to introduce you. You can go to sleep, Monica.
Okay.
For a while. So gives me absolute pleasure to introduce Dr. Monica Gramatges to our ICOS seminar series. Monica and I have known each other for donkey's years. She's been giving me a dime per conversation that we've had, and I have a jar full of those, right, Monica?
Right.
Right. Monica did her med school from the University of Texas Medical School, went on to do her residency in pediatrics at Oregon Health and Sciences University, and then she went to do her fellowship at Stanford University. And then she came back to Houston and decided that's where she wanted to be. She is now an associate professor of pediatrics. I've known Monica in terms of her desire to take the molecular underpinnings of treatment-related complications and build upon that. And she stayed very focused on that aspect. She's recently won several awards and honors. I'm going to highlight two of them. One of them was the American Society of Pediatric Hematology Oncology Childhood Cancer Survivorship Excellence Award. And then, which is going to happen this year in May.
Mm-hmm.
That's going to be fantastic. Is it going to be in person? No.
No, it's virtual. Mm-hmm.
Okay. And then she's also been selected through a very competitive process, I know that, for the ASCO Leadership Development Program. So that's another amazing journey that she will undertake this year, starting this year for a full year. So as I had said, her research focuses on molecular aspects of cancer therapy-related toxicities. What I omitted to tell you also is that she's truly a physician scientist. She not only takes care of patients, but uses her PhD to the full extent and has done a great job in teasing apart the actual cellular and molecular level of how telomeres influence childhood cancer survivors' complications, in particular second cancers. And so with that, I'm going to give it to you, Monica, and have you take it from here, okay?
Okay. Thank you so much, Smita. That was such a sweet introduction, and I don't know if I deserve it, but I'll try to do your introduction justice and and give you all a nice talk. So I wanted to talk today about the results of a project I've been working on for several years now. I'm at the end of an R01 funded project to investigate telomere length and risk for thyroid second cancers. So, and let me just forward, let's see, make sure I can forward my slides. Hold on, I'm having minor problems with this. There we go. Okay. So, and I should say I have nothing to disclose. So this is our hospital, Texas Children's. All of the faculty at Texas Children's are faculty at Baylor College of Medicine, essentially. So Texas Children's is the pediatric hospital, and you can see it. I don't know if you can see my pointer, but it's here is our main hospital and clinics. And the objectives of my talk today will first be to discuss the risk determination of late effects in childhood cancer survivors. And I'm going to focus on subsequent malignant neoplasms and thyroid second cancers. I will review biomarkers of aging in survivors of childhood cancer and focus there on telomeres and telomerase. And then I'll present the results of our R01 funded research that assess the relationship between telomere length and risk for thyroid second cancers. And last, I'll discuss the translational relevance of these findings and then implications, at least how I see it, for survivorship research. So to introduce this talk, I'm just going to briefly review, and I think all of you probably are well aware of this, but we now know that while we've been able to cure the majority of childhood cancers, 1/3 still develop a severe, disabling, or life-threatening chronic health condition within 20 years, including therapy. Fortunately, that risk has declined with subsequent treatment eras. So more survivors or children who are treated more recently seem to be at less risk for these chronic health conditions, but they're still dramatically at higher risk for these issues than sibling controls. This is from the Childhood Cancer Survivor Study in a nice paper from Todd Gibson and the CCSS group. So the risk for late effects is determined by treatment exposure and dose, and then further moderated by genetic susceptibility, demographics, socioeconomic factors, and behavioral choices. So all these things come together to produce risk for late effects, and there are a number of modifiable factors over time that contribute to that risk. So one of those late effects, the subsequent malignant neoplasms, and we know that the absolute risk increases with increasing age. And after the age of 40, the standardized incidence ratio for survivors is at least twice expected. Most subsequent neoplasms develop in previously irradiated sites, but chemotherapy alone is also a risk. And the most common second cancers are breast, sarcoma, thyroid, colorectal, CNS, non-melanoma skin cancer, salivary gland tumors, and leukemia. So the Childhood Cancer Survivor Study, and I'm sure many of you know, is a retrospective cohort study with longitudinal follow-up of 5-year survivors of childhood cancer that were diagnosed at multiple institutions in the US or Canada in the 70s through the 90s. Radiation dose in field and chemotherapy agents and dosing for select agents were collected from all participants. And this cohort has been prospectively followed now for decades to ascertain late effects of their cancer treatment. For this study, subsequent neoplasms were identified via self or next of kin proxy report or death certificates, and then confirmed by CCSS designated pathologists or when unavailable, death certificates and medical records. And the study includes only people who survived at least 5 years after their initial cancer diagnosis. The highest risk for second cancers in CCSS is among Hodgkin's lymphoma and Ewing sarcoma survivors. And this is a graph from Deb Friedman's paper from 2010 that looks at the cumulative incidence of SMN at 30 years, and it was about 8%. If you look at this graph, and that's among nearly 15,000 survivors. And you can see to the right that radiation therapy dramatically increases that risk. So there is now evidence that genetics play an important role in risk determination of cancers. And we've now seen several really lovely papers come out demonstrating risk, for, mutation risk for second cancers. So from the St. Jude Life group, Zhao Ming recently showed that rare pathogenic mutations in cancer predisposition genes occur in less than 10% of survivors. It's rare, but it is there. And these are mutations in genes like TP53, and they may be associated with breast sarcoma or radiation-related second cancers in particular. GWAS studies have also identified risk loci for AML, breast cancer, and basal cell carcinoma. And Lindsay Morton just recently, several months ago, produced a really nice paper that showed radiation-related second cancer risk is associated with rare variants in DNA double-strand break repair genes, which is super interesting and I think something we all suspected, but she was able to show this very nicely in this JCO Precision Oncology paper. So radiation and risk for second cancers, and just to briefly introduce, for the most part, for most second cancers, is a linear dose response relationship across all dose levels. The more radiation you get, the higher your risk is for breast, brain, lung cancer, and sarcoma. With thyroid cancer, it's a little bit different. There's a linear exponential dose response with plateau or downturn in risk at doses that are above 20 to 30 gray. And that is thought to be due to just the radiation cell-mediated kill. The relative risk for thyroid cancer is, there we go, thyroid cancer. The radiation dose response relationship, as I explained, is a flattening of risk at higher doses. And this was first noted in the 1990s. And the Childhood Cancer Survivor Study showed this about a decade later, demonstrating a linear increase in risk up to 20 gray, followed by a downturn with excess risk among survivors who were exposed at a younger age, females, and longer time since exposure. There's also an indication of increased risk associated with prior treatment with alkylating agents, in particular procarbazine, but not to the same extent as with radiation. And these two graphs are from the Sigurdsson paper, which is the CCSS paper in the Lancet, just demonstrating that downturn in risk around 20 to 30 gray, and then the younger age at diagnosis being associated with a higher relative risk. And we've seen changes in risk for SMN over time. So risk stratified therapy and a decline in chemotherapy, and particularly radiation dose over time, has led to a decrease in second cancers for survivors who are treated in more recent decades. And this is true for all SMNs. And if for all SMNs, this seems to be driven primarily by a decline in breast cancer and soft tissue sarcoma incidence. But if you look at thyroid, it's a little bit different. And I wanted to bring this up because most of my talk is going to focus on thyroid second cancers. Then in Lucy's paper in JAMA from 2017, thyroid cancer seemed to increase in incidence in more recent treatment eras. And we think that effect is related to increased detection of these indolent, slow-growing nodules, because thyroid cancer is generally a slow-growing cancer. And that's been prompted by more aggressive surveillance strategies. So basically, we're finding it more because we're more aggressively surveilling survivors for that outcome. Second cancer risk assessment and surveillance recommendations, as we all know, are based on survivor demographics and data that's obtained during childhood. So when you have the long-term follow-up guidelines, we're using patient information, for example, their sex, their age at diagnosis, and the exposures they receive, the dose of chemotherapy, the radiation fields, to estimate their risk for late effects like second cancers. We are taking into account to some degree factors that occur over time, like if, for example, for breast cancer, the age they achieved menopause, or whether or not a woman was childbearing. But we know that there's a number of potential behavioral and lifestyle factors that can moderate this risk, that are much harder to assess or take into account over the lifetime of the survivor. So this risk can be modified by both genetic factors and specific events and lifestyle behavioral choices. And this is a figure from Lucy's paper indicating that, that phenomenon. So I felt as though there's a need to identify biologic and molecular markers that measure and assess the impact of environmental contributors to risk for SMN, and of course, other late effects. And that's really been the large focus of my research in survivorship. So here you see the same figure from before with the child that was exposed to radiation and chemotherapy, and the genetic factors that, and the genetic contributing factors that lead to an outcome or late effect as that child grows into an adult. But what's to say that that same child wouldn't have a much more significant aging effect from cancer or more severe late effects? And how can we best assess the impact of these lifestyle choices and changes and environmental contributors to cancer? Are there differential responses to chemotherapy and radiation that vary from individual to individual? I feel as though a biomarker that suggests heightened risk could facilitate targeting specific and at highest risk population for interventions. So to talk a little bit about what these biomarkers would look like, we know now that, well, there are biological hallmarks of aging that have been demonstrated for decades now in elderly populations. And more recently, in work that has been largely led by Curie Ness and collaborators at St. Jude, have really shown that many of these biomarkers hold true for childhood cancer survivors. So not only are survivors more at risk for frailty, sarcopenia, or early onset aging phenotype, but there's now a number of papers that have suggested they also have underlying biological indicators of early onset aging. And those include evidence for genomic instability, excess DNA methylation aging. There was a nice paper from St. Jude about that recently. Loss of proteostasis, mitochondrial dysfunction, systemic inflammation, and last, excess telomere shortening, which I'm going to talk more about now. I think, there it goes. So telomere length shortens with DNA replication in cells that have been sufficient to telomerase. What are telomeres? So telomeres are the conserved repetitive DNA protein structures that are chromosome ends They're kind of like, I like to say, the plastic tips and the ends of shoelaces. They protect chromosome ends and protect chromosome integrity by preventing the loss of terminal sequences during DNA replication and aberrant DNA repair activities like chromosome end-to-end fusions. Telomere length is genetically determined, so we inherit it from our parents, mostly our dad, and in cells that don't express telomerase. Telomere length shortens with each cell division, and progressive shortening continues until telomeres become critically short, which is known as the Hayflick limit, and that induces cellular senescence. So peripheral blood telomere length shortening over time can be impacted by environmental stressors. So everybody's telomeres shorten. Some people shorten faster than others. And factors that have been shown to augment the rate of telomere shortening over time include prolonged anxiety, depression, or stress, and higher BMI, so obesity, smoking, and now recently even space travel apparently is not good for telomeres. Telomerase is the principal mechanism for telomere maintenance and extension. So what I'm showing here in this figure on the right, on the top right, is the enzyme complex of telomerase that's primarily expressed in germ cells and stem cell populations. It's a reverse transcriptase. That is the principal mechanism by which telomeres are maintained and extended, but it doesn't do this alone. So telomerase cooperates with a number of other proteins that act at the telomere to maintain telomere homeostasis. And mutations in any one of those genes that code for these proteins can lead to excess telomere shortening, or in some cases, like for example, POT1, very long telomeres. And these findings are often associated with a distinct clinical phenotype. If any of you work on bone marrow failure, an example is dyskeratosis congenita. So what's important for the remainder of my talk is to talk a little bit about how telomere length is measured, because I'm going to talk about that in the next coming slides. So telomere length can be estimated using a variety of techniques. I'm just going to talk about 3 of them. So telomere restriction fragments, TRF, and that's a teloblot, visualizes telomeric DNA using a radioactive telomere probe. And that's historically been considered the gold standard, but it's a very slow technique, it's time consuming, and it requires a considerable amount of fairly high quality DNA. In contrast, what my lab uses is both qPCR, and we collaborate with Repeat Diagnostics to use telomere flowfish to quantify telomere length. Quantitative PCR amplifies telomere DNA relative to a single copy gene and is a high throughput and very inexpensive method. It requires very little DNA, and it's useful in case control comparisons, but it doesn't provide an absolute length. So you really, it's best utilized when you have 2 large groups of patients and you're comparing them. Flowfish, or telomere flow fluorescence cytometry, requires fresh or viably frozen leukocytes. It is used exclusively in leukocyte cells. It is considerably more expensive, but it does produce an absolute telomere length. And even more importantly, the Repeat Diagnostics group uses this test, or applies this test clinically as a diagnostic tool for telomere biology disorders such as dyskeratosis congenita. So they've actually developed a standardized curve from close to 1,000 patients, which has led them to be able to develop these percentiles. So the expected median telomere length for each given age. They collected samples from individuals aged 0 to 90, and then developed percentile curves so that when you order this as a diagnostic test, your results return, like shown in the figure there, as a point plotted by your patient's age on the percentile curve, so you can tell where that patient falls on that percentile curve. And this is an example of telomere flowfish output data without the percentile curves, just demonstrating that telomere length shortens over time in granulocytes and lymphocytes to different degrees. So leukocyte telomere length really serves as a molecular clock for cellular replicative aging, and that's shown here. Telomere length is shown, the age is shown in years across the bottom, and telomere length in KB on the y-axis. And the decline that you see later in life, like after age 70, 80, reflects the well-described phenomenon of aging-related deterioration in the immune system that results in impaired responses to infection, excess risk for cancer, poor response to vaccination, and low levels of systemic inflammation. It's all this immunosenescence that occurs with aging. So that's reflected in shorter telomere length in elderly populations. And we know that peripheral blood telomere length is associated with cancer risk. This is a known phenomenon of aging as an excess risk for cancer, but there have also been a number of population studies that have looked at peripheral blood telomere length as a predictor for cancer. 2 meta-analyses showed an overall increased risk for cancer with short blood telomere length. And then a third, more recent study was restricted only to population studies and looked at all the different kinds of cancers. And this is primary cancers in the general population, mostly in adults, and really showed that the risk is driven by gastrointestinal and head and neck cancers in terms of short blood telomere length and excess risk. So what's the relationship between telomere length and risk for second cancers? Most studies conducted to date have studied telomere length in relation to risk for first cancers. So back when I came to Baylor and I met Smita for the first time, and we talked about this idea, and I made my first proposal to the childhood cancer survivor study to access samples and data. This was my question. In cancer patients, I knew that in cancer patients, there have been a number of studies that showed that chemotherapy and radiation lead to short and long-term telomere shortening. And there have been various pathways proposed, like DNA damage and heightened proliferative demand for hematopoietic reconstitution. And then generally, there's been more substantial data with radiation exposure. But what I really wanted to know was what about second cancers? So if telomere length predicts first cancers, then what about survivors of childhood cancer? Is there an association between telomere length and risk for second cancers? So we looked and we hypothesized that survivors with radiation-related second cancers would have shorter telomere length than survivors without second cancers. And we conducted this study in the Childhood Cancer Survivor Study. And this was a long time ago. I did it when I first came to Baylor. I was under an institutional K-12 when this was part of my K-12 project. And we compared survivors without second cancers to those with second cancers and matched them by primary diagnosis, age when the sample was collected, the length of time between primary diagnosis and sample collection, and then their chemoradiation exposures. And what we found was that compared to survivors without second cancers, survivors with radiation-related second cancers had shorter telomeres. And we also noted that that association was really driven by thyroid second cancers. So that prompted us to then follow up this paper with the proposal for a larger research study to investigate the molecular mechanisms for telomere shortening in survivors of childhood cancer who develop thyroid second cancers. And this was funded as an R01. And the initial hypotheses, or the first aim of this project, was to ask if common genetic variation distinguishes survivors with thyroid second cancer from those without second cancers. And our first hypothesis was that survivors with thyroid second cancer would have shorter genetically predicted telomere length, which would be determined by a polygenic risk score, suggesting shorter telomeres at baseline than survivors without second cancers. And our second hypothesis was that survivors with thyroid second cancer would be more likely to have common pathogenic variants in telomere biology disorder genes than survivors without second cancer. And for that, we did a functional variate assessment. So to do this, we again collaborated with the Childhood Cancer Survivor Study, which I've already described a little bit, but I'll just state that thyroid second cancers from the CCSS are defined as any second cancer of the thyroid gland that occurs as the first second cancer with a diagnosis and date of diagnosis verified from the original pathology report by a CCSS designated pathologist. I should also mention that buccal, saliva, and peripheral blood samples have been collected from a subset of CCSS participants, and they've now completed genotyping that has been conducted from the buccal and saliva samples using the Illumina Human Omni5 exome array, and that was done by the NCI in collaboration with the NCI. So for the first hypothesis that survivors with thyroid second cancers have shorter genetically predicted telomere length, we used a polygenic risk score approach. So we determined from published GWAS a list of SNPs that were related to telomere length in the general population. And then we looked at both individual and combined SNP associations with thyroid second cancer, which led us to an unweighted and a weighted estimated hazard ratios based on the number of risk alleles present and the strongest published effect for each allele. So we had 118 cases for this study and over 5,000 controls, and we had identified 9 common SNPs that were previously associated with leukocyte telomere length by genome-wide meta-analyses. We evaluated the association with risk for thyroid second cancer for each individual SNP, as well as weighted and unweighted genetic risk score and adjusted for known covariates. And we showed that variation in common SNPs influencing leukocyte telomere length is not associated with thyroid second cancers. And that was including considering the total burden of the effect alleles for these SNPs on thyroid second cancers. And we saw no relationship for either the unweighted or weighted genomic risk score. We also restricted the analyses to only to radiation exposed individuals and still observed no associations. This was published as a null report in CEBP. And then for the second hypothesis, we took a functional variant approach, and we determined a list of genes that were implicated in disorders of telomere maintenance or risk for familial cancer. And then we refined that data set to include only functional common SNPs likely to affect gene transcription, and estimated the unadjusted and adjusted hazard ratios for each SNP. And this, again, the purpose of this was our hypothesis was that survivors with thyroid second cancer would be more likely to have common pathogenic variants in telomere biology disorder genes than survivors without second cancers. So again, we used the CCSS. We had 110 cases for this study and close to 5,000 controls. And we included SNPs that were within 100 base pairs of 15 telomere biology disease disorders, and they were annotated using Regulome-DB. So Regulome-DB is a database that annotates SNPs with known and predicted regulatory elements in the intergenic regions of Homo sapiens genome. And it generates a score that confers the projected functional impact of each variant. So for this study, we included SNPs that scored less than or equal to 2, which signified a high likelihood for affecting transcriptional regulation. And then we conducted a time to event Cox regression for thyroid second cancers, evaluating the risk for thyroid SMN for each functional SNP, and adjusted for demographic and clinical factors. For lower frequency variants, we also validated our results of the Fisher's exact test. This is just a list of the genes that we included in case anyone is interested. And they are all genes that are either related to telomere biology disorders or have been shown to be impacted in familial cancers and in association with telomere shortening. And what we showed from this study, we found that the minor allele at the protection of telomeres 1 or POT1 gene was associated with risk for thyroid SMN. We found 60 SNPs that were located in or near telomere candidates that met our inclusion criteria, but only one, this variant in POT1, met the Bonferroni criteria for statistical significance. And it was present in a relatively small number of cases and controls, 3 out of 110 cases and 14 out of 5,000 controls. So definitely not the answer explaining what is the risk association, but is certainly an interesting finding. And then we decided, because we knew that there was recent data that mutation-induced disruptions in the POT1 TPP1 complex, which are both components of shelterin, which is important for telomere homeostasis, affected that ability of the complex to bind telomeric DNA and led to longer telomeres and more fragile telomeres that may promote genomic instability and lead to cancer risk. And that there have been several papers published on germline variants in POT1 in association with various cancers, including like familial glioma, familial melanoma, colorectal, ovarian, lung cancer, all these studies and cancers with germline variants in POT1 and longer telomeres and the cancer risks. And we thought that was really interesting, so we decided to look. We actually were able to show that our survivors with the POT1 variants had longer blood telomere length than survivors without the variant. Not long enough to be like a diagnostic test, but certainly kind of fills what you would expect with a POT1 variant. All the survivors with the variant had leukocyte telomere length that was at or above the 25th percentile for the most part. And B cell telomere length was particularly interestingly longer in the top 10th percentile in survivors with the variant versus with those without the variant. And that was significantly different. So to sum up this part of the talk, that common genetic variation is not a substantial contributor to telomere length shortening observed in survivors of childhood cancer who develop thyroid second cancer. So then our next phase of this project, the second aim, was to really ask about what about rare variants? So we looked at the common variants, but are survivors with thyroid second cancer more likely to have rare pathogenic variants in telomere biology genes than survivors without second cancer or even non-cancer controls? So our third and fourth hypothesis were that the survivors with thyroid second cancer would have a higher frequency of rare variants with large effect sizes than survivors without second cancer and non-cancer controls, and that survivors with thyroid second cancer would have a shorter telomere length in leukocyte subsets than survivors without second cancer, using telomere length by flowfish as a proxy for functional analyses of mutations that occur outside of the biology genes that we tested. So again, this project was done in collaboration with the Childhood Cancer Survivor Study. We were able to identify 52 cases with thyroid second cancer matched with 52 controls that had no second cancer. This was a matched pair analysis using viably frozen blood samples from CCSS, and of course, excluding bone marrow transplant recipients because they don't have their germline blood anymore. The matching criteria were by primary diagnosis, the year since primary diagnosis, chemotherapy, yes or no, the radiation fields, everyone got radiation on this study, and the duration of follow-up. So we also conducted whole genome sequencing for this project. We included pathogenic or likely pathogenic variants, or those with a minor allele frequency of less than 1%, and restricted, and those are pathogenic or likely pathogenic as designated in ClinVar, which is a database of those kinds of variants, and then restricted to exon intron regions and telomere biology disorder genes. The telomere length we determined from viably frozen bank samples by telomere flow fish in collaboration with Repeat Diagnostics in Vancouver, British Columbia. We measured absolute telomere length measured in leukocyte subsets, and the results were then transformed to age-adjusted percentiles. So of the survivor pairs, we had 46 or 92 total survivors that had sufficient cell recovery for flowfish, which is pretty impressive. It took some time to get there. We had to replace several samples, but we got a good amount. 88 of them had sufficient DNA for whole genome sequencing. And what I'm showing on the left is just a comparison of the survivors with and without second cancer by mean age at blood sample collection, and mean time from first cancer diagnosis to blood sample collection. And then we had 20 pairs that had Hodgkin's lymphoma, 13 with ALL, 7 with CNS tumors, et cetera. All of them received radiation, and then 35 pairs also received chemotherapy. So we first showed that rare variants in telomere biology disorder genes are more common among survivors than controls. So we found rare variants in coding regions in 23 survivors, 14 that had second cancer and 9 that did not have second cancer. None of the variants that we found were pathogenic. One of the variants had been previously reported in association with telomere biology disorders, but is, so far as we can tell in our own lab and in other labs, does not seem to impact telomerase function. And if anyone's interested, I can talk more about that variant at the end. All of the subjects with variants were heterogeneous. 5 out of 8 survivors had a missense variant in telomerase reverse transcriptase, or TERT. And we had previously sequenced TERT using similar whole genome sequencing technologies and non-cancer controls. So we compared them to see out of interest if the survivors seem to be enriched for variants in telomerase reverse transcriptase. And the survivors were almost 4 times more likely to have a rare TERT variant than non-survivor controls. This wasn't statistically significant and not part of our original aim, but I included here just for perspective on what the incidence of the variants means. These are the variants that we found. They all have a minor allele frequency of less than 1%. It's kind of a busy table. I just kind of wanted to give an overview of the genes where we found variants. And you can see the total number of survivors, 14 with thyroid second cancers and 9 without second cancers. There were a few variants that occurred in the same survivor, which is interesting. We use the whole genome sequencing data to determine telomere length, which can be done through software like Telomere Cat. This was simply done to verify and show that there was an inverse relationship between the whole genome sequencing determined telomere length and age, and also because we wanted to know, we wanted to compare survivors with thyroid second cancer and those without second cancer, and it does appear as though the relationship is more pronounced among survivors with thyroid second cancer. I think this graph is a little bit misleading because it implies that they're shortening over time, and I just wanted to remind everyone we had one time point from each of these people, so really just comparing the telomere length obtained from that sample from that one time point and not showing a trajectory over time. But it is still supportive of of what we'd expect to find, which is that telomere shortens with increasing age. So these are the flowfish data. So when you send blood samples to repeat diagnostics, this is the output that you typically see. And what they provide, you can ask for either a single panel of lymphocytes only, or you can ask for the multi-panel that includes naive T cells, B cells, NK cells, and memory T cells. And what I'm showing here are the survivors with second cancer in red and the survivors without second cancers in black. And on first glance, you look at this and you think, okay, so they're kind of all hovering in the middle there, like what you'd expect. You can see our survivors between the ages of 20 and almost 60 years old. But what's interesting about this, and what I think is super important, and if any of you treat bone marrow failure patients, you'll recognize this right away, that it is super weird to see this many people who are below the first percentile. So I show here on the red line, that represents the first percentile for age. That means less than, or 1% or less of the general population of telomere lengths that short. And look at our survivors. There's a great number of them that are below the 10th percentile, way more than there should be. And if you look at Blanche Alter's initial work in dyskeratosis congenita, where she took patients with that telomere biology disorder and compared them against their parent controls using telomere flowfish, you can clearly see that patients with dyskeratosis congenita, shown in color in the boxed graph on the right, fall well below the first percentile, to the point that telomere flowfish is actually used as a diagnostic test for dyskeratosis congenita. Our survivors, many of our survivors look like dyskeratosis congenita patients. And I just think that is really interesting. We had about 8% of our survivors had lymphocyte telomere length less than the first percentile, and 30% have lymphocyte telomere length less than the 10th percentile for age. So way more than there should be. And I'll show that in another graph in a minute. And we also found that survivors with thyroid second cancer had shorter absolute telomere length than those without thyroid second cancer. So we conducted a matched pair analysis. And the difference between survivor median telomere length and the population median, so like how we get the percentiles, was much larger for the cases with thyroid second cancer than it was for control. They were deviated further below the norm. And that difference reached statistical significance in 4 out of 5 leukocyte subsets. 8 survivors, all were cases with thyroid second cancer, had an NK cell telomere length less than the 10th percentile. And 22 cases and 5 controls had naive T cell telomere length that was less than the 10th percentile. So the cases were almost 3 times more likely to have naive T cell telomere length and less than the 10th percentile than controls. And that was significant. And I show there for each subset the deviation from the age-based population median for those with and without thyroid second cancer and the p-values. The other thing to note is that the shorter telomere length that we observed in naive T cells corresponds with a lower naive T cell population frequency. So naive T cells actually comprise a smaller proportion of all lymphocytes in cases versus controls. And what we think this means is that the shorter telomeres that are seen in the cases induce cellular senescence. So these cells are no longer around, like they just have less of them, the survivors who have thyroid second cancer. And then to show this kind of more clearly, so we built this graph so the survivors with thyroid second cancers are shown on the left for each color pair. They're kind of the lighter color, and the survivors without second cancers are on the right for each pair. And this is by cell type, so B cell lymphocytes, memory T, NK cell, naive T cells. And on the y-axis is the age standardized deviation in telomere length. And what you see here on the red line, that's like the population median. So you can see they're all sinking. Like if you imagine the red is the water level, they're all sinking below. They're all below the norm. But the survivors with thyroid second cancers are sinking more. So I think for me, this was a helpful visual to really understand what was happening. And these violin plots are very illustrative in that sense, that you can see where the distribution of the telomere length falls for each leukocyte subset. So what's the translational relevance for this? And I'm close to being finished. So we know that adaptive and innate immunity decline with aging. And this is due to a decline in naive T cells, reduction in T cell receptor diversity, decline in NK cell function. I am not an immunologist, but I find it fascinating that our survivors are demonstrating signs of aging in their immunity, and at least in their telomere length in their leukocytes and in their cell populations. We've shown now that survivors of childhood cancer have shorter than expected telomere length, nearly 25% with telomere length less than the 10th percentile, and that survivors with thyroid second cancer have shorter telomeres than survivors without second cancers. This effect is clearly more pronounced in naive T cells and NK cells. And what's interesting about that is that those cell types are important in cancer surveillance. So my next hypothesis is that some long-term cancer survivors experience immune dysfunction, and that includes impaired cancer surveillance, which may confer a heightened risk for second cancers. And the super interesting thing about this is that in the 1990s, Crystal Makel and others since her showed that cancer chemotherapy and radiation deplete naive T cells and NK cells. So we know this happens during cancer chemotherapy. And studies have even been extended up to 18 months post-cancer therapy, showing this effect, and also showing an impaired DNA damage response in children receiving cancer chemotherapy. But to my knowledge, no one's ever looked in survivors this old, this far out from treatment, to see what the long-term impact on immune function is. I mean, we all suspect it and know it. We know survivors may not respond as well to vaccination. We know that they may be at heightened risk for infections, but the immune function studies haven't yet been done. And I think that's something that really is, this really does beg the question, what is the immune system doing? What's happening? And is this really truly premature immunosenescence that we see in elderly populations? And the last thing I want to share is that I do think there's some implications for screening here as well. So there are recently released recommendations from the International Late Effects of Childhood Cancer Guidelines Harmonization Group that includes screening at-risk survivors with either neck palpation every 1 to 2 years or ultrasound every 3 to 4 years. And I wanted to remind everyone that telomere FLISH is actually being used as a clinical diagnostic test for disc keratosis congenita. And we know that lymphocyte telomere length has an 85% positive predictive value for correctly making that diagnosis. And what I'm wondering is, perhaps NK cell telomere length, which has a very high specificity for thyroid SMN in this small sample set, may be useful as an adjunct to guide clinical decision-making prior to surgical intervention. Like you find a nodule and you're trying to decide whether to do an FNA or whether to do a more extensive surgery. This might be a useful tool to help with that, and obviously needs to be studied in larger populations and prospectively. So then just to tie up the future directions, I do think that these findings warrant an expansion of the analysis to assess generalizability to other second cancers. I would love to look at telomere length shortening over time through prospective serial assessment, because that hasn't been done, and I really want to know what the trajectory is over time and what is the slope of that curve. In conjunction with in vitro and clinical measures of functional immunity, which would require me to find an immunologist collaborator. And then given the lack of baseline and pre-chemo radiation telomere length assessment, because we don't really have those peripheral blood viably frozen bank samples from patients before they're diagnosed, at least yet, I would like to correlate the functional immunity and actual telomere length with genetically predicted telomere length as a proxy, since we don't really have a great way of doing that, to understand what is the baseline and how much is the baseline telomere length impacting these outcomes. And then last, as I mentioned, prospectively test the utility of telomere FLOWFISH and thyroid second cancer surveillance. And with that, I'll finish and acknowledge I have a very small lab, and I do a lot of my work in collaboration with outside investigators. We have a wonderful epidemiology program with Philip Lupo and Michael Schur and Melissa Richard, and I work with them quite frequently. And then, of course, I have wonderful mentors, Smita, and also my local mentor, Allison Bertouche, who's a bone marrow failure doctor who specializes in dyskeratosis congenita. So a lot of my early research and work in her lab was looking at AML and telomere biology disorders and telomerase defects and implications for telomere biology. I collaborate extensively with Geraldine Albert at Repeat Diagnostics and our local FlowCore, and my funding is listed there as well. And of course, I'd have to acknowledge the Childhood Cancer Survivor Study, without which none of this work would be possible. And I will close and take questions.
Thank you so much, Monica. That was just absolutely fantastic. So I'm opening this up to everyone for questions. And while we're waiting, I'm going to ask you, Monica, something that I'm sure you've been asked before, that we're measuring telomere lengths in the peripheral blood. And often we're mashing up all the leukocytes together and measuring, because if it's pre-collected DNA, extracted DNA, we haven't sorted them if you're doing qPCR. So how does that correlate with the organ of interest? So for example, if somebody were to say that I want to see if there's an association between tumor length and cognitive outcomes, without knowing whether we're measuring telomere length in the glial cells, how would one?
So not all tissue types have been assessed, but there is a very nice paper. The first author is Gadala. She's a mentee of Sharon Savage. Now she's probably has her own lab and does, but it's from several years ago. And she assessed telomere length in at least 4, maybe 5 tissue types, and showed that there's strong intra-individual correlation. There is some discrepancy by tissue type, but you can see different individuals definitely stick together. So there's strong individual correlation between tissues. And she was looking at fibroblasts and blood and some other, I think she had several tissue types in that study, and I don't recall exactly, but I've looked to that as evidence for the ability of using telomere length as a proxy for kind of whole body telomere length. I think the blood telomere length poses, it's a very easy tissue type to collect and assess. And so largely the studies that I've seen have used that as a method. I haven't myself. I've been asked about what about thyroid tissue? And, I think that's a whole different question, what's going on in the cancer itself than what's going on in your immune system. And I think for this work in particular, you're right, the qPCR is flawed. It's kind of a very rough, I think, it's almost worked out well the way we used it, right? Because it was kind of the first way that kind of opened up, okay, so there's this relationship, but it doesn't really help you understand any of the details. And I think the flowfish is just a really nice way to know what's happening in each subset and kind of hone in on where the problems are. And the flowfish study for this work, for example, with the strong associations we saw in naive T cells and in K cells, really made sense, but we would never have known that if we hadn't assessed with using flowfish. So I'm not sure if that answered your question, but I can tell you from the psychological outcomes, there are a number of studies, and some of our local investigators are interested in this, that examine telomere shortening in association with anxiety, stress, depression. There are large epidemiological, population-based studies that are doing that and have shown associations there, even though they're not measuring what's going on in the glial cells specifically. So I think there is room for that exploration.
Good.
Sure.
Smith, there's a question in the chat box. Smith, do you want to ask it yourself?
Oh, you're muted.
Thank you. That was great to listen to your talk. Very inspirational for all of us junior investigators. My question is about if one had limited resources, what would be the tissue to store? And why do you think there is so much heterogeneity across tissue types in the same individual in terms of telomere length?
Well, so there's differential telomerase expression by cell type. So some cells express more than others, and that affects telomere homeostasis. So I think that's why there's tissue differences in telomere length. I mean, we all kind of start at the same starting point, but then over time, depending on which tissue you're measuring, there may be differences in the rates of attrition. But again, what's important, I think, is that there is that intra-individual correlation that you can at least use to justify your use of banking, for example, blood. I think the easiest tissue type to bank is blood. And what I do now, and what I've talked with our epidemiology team about doing, which I think is so important, is thinking ahead. I mean, sample banks are so important to establish early and do it the way you do it right. And we've really made it a mission to bank viably frozen leukocytes, like to viably freeze leukocytes in our sample banking, just because it provides you so much more flexibility. I mean, if you want to use it for DNA, you can, but you also have so much more opportunities and things you can do with those cells than you could if you just blanked a blood sample and extract the DNA. So I mean, at our tissue bank here, we include blood that is then used for both freezing leukocytes and also for DNA extraction. And then we also bank plasma. And then we bank CSF on all of our patients and even urine. And in those who get bone marrow aspirations and biopsies, we get fibroblasts because it's so easy to do when you do the bone marrow test.
Good.
And it's become a really rich resource for so many lines of research. I mean, just having that access to that has been transformative.
Noha.
Yes, yeah. First of all, thank you so much for a wonderful talk. It was very, very interesting. You know, just to piggyback on everything that's been said, and going back to polygenic risk score, how you built that, you relied on the GWASs from a general population to build it. And I was wondering if everything was said in terms of, you know, selecting those variants to build your PRS and how that might have affected not seeing that association as well, what thresholds been used to build that?
That's a great question. And I should really acknowledge the team at the NCI who are collaborators on that project. Mitch and Lindsay Morton, they were very helpful in consultation with how to approach that, because it was the first time I'd really written a polygenic risk score paper. So I really relied on others' expertise quite a bit. And what we ended up using, they're actually, fascinatingly, and something I didn't know before I embarked on that project, there are now like a number of papers that have used polygenic risk scores for telomere length in association with various outcomes. And it's like the conventional wisdom accepts these sets of 9 genes. So there's been some modification over time where there's like, you know, there'll be 8, there'll be 9, there'll be 11. So there's some like, but it seems as there was an accepted approach at the time that we took. And since then has evolved, and now there are additional markers that would be used. So it might be worth reapproaching that using the more expanded gene panel now, because when we did that paper, these were the 9. And that was like everyone, you know, they really felt like the NCI team really felt like this is the accepted approach. This is what we should use. But it is probably worth revisiting it with the additional genes and variants that are used now for that very reason.
Yes, I mean, exactly that. It's considering how comprehensive it is, but also considering the setting where you're testing, like you're in the setting of secondary cancers. So you would need to consider how that genetic risk plays in that setting versus in the general population, if that makes sense.
Yeah, that's true. That's true. Thank you. That's a great question.
Any other questions?
So Wendy, we did actually show in that last set of data that I presented that telomere length predicted mortality. So shorter telomeres predict mortality. I didn't include that in my presentation because Les Robeson has reviewed my paper. He did not like the data and thought that it wasn't as convincing as it should be. So I left it out of my talk. But Sven and I can talk more about that. But it was significantly associated with mortality. And that my study is only 50, 90 odd patients. It's very small sample size. There have been a number of larger population studies showing shorter telomere length and risk for mortality. So I think that is worth exploring, but would need to be done probably in a larger sample size. Yeah, I mean, I will say the frailty story is a sad one because Carrie and I have submitted that grant now 4 times and it is yet to be discussed. And I think it's an awesome grant and people are not convinced. The reviewers are not convinced that frailty is a thing in survivors. And I've been trying to convince them for years and I can't seem to convince them. But that grant proposes in childhood cancer survivor study serial assessments over time, going back, finding survivors, collecting samples serially at 3 time points to really understand what the trajectory is like. Is it a flat line? Is it, how is it linear? Is it nonlinear? What's going on? And looking not only at telomere length, but also DNA methylation aging, and we proposed inflammation as well. So I would love to do that study. I just need someone to give me the money to do it.
So I'm going to take this question back up because, frailty is a big thing and accelerated aging and especially in our older patient population. So now I'm talking on behalf of some of our geriatric oncologists that are part of our institute. And, there's heterogeneity there in terms of who is aging faster than the other group. And we're trying to address this by seeing if interventions such as those offered by physical activity or diet can improve certain aspects of frailty. So I'm going to ask you this question is that suppose there were a relationship, a tight correlation between telomere length and frailty, and we intervened with physical activity and diet, would we see anything in terms of telomere length?
So,
or just the trajectory of it?
The experience to date indicates that you see an early response with intervention with telomere length, but it's not durable. So you have to maintain the activity over time. And as soon as your intervention stops, telomere length reverts to where it was before you started. So I don't know. I think it may be more useful in identifying high risk populations that might benefit from an intervention. But I don't think the telomere length itself is necessarily important. I think in terms of like, if it continues to shorten, I don't know if that means anything, but I think it may mean something just to identify the population at risk, right? That's kind of how I think of it. I may be wrong, but that's kind of how.
Well, I think there's much work to be done there.
Yeah.
To figure that out.
Yeah, I think so too.
Yeah. But let's all go for a walk now.
Okay.
Monica, that was absolutely amazing. So
thank you so much for the invitation. I sincerely appreciate it. And I just really enjoyed getting a chance to talk to all of you. And of course, if anyone would like to email me or has any follow-up questions, I'm more than happy to answer them. And I love to collaborate. So if you have great ideas, let me know.
Monica is our absolute, if there's any question on telomere lengths or telomere homeostasis, we can go to Monica. So thank you.
Thank you.
Thank you so much.
Thank you.
Bye.
Take care. Bye-bye.